Hi, this is Dr. Collins Jones. Welcome to segment 3 of my video series on manual cell counting and cell density calculations. In this segment, we will cover actual cell counting using the hemocytometer. Let's pick up where we left off in segment 2. So we're assuming the hemocytometer is properly loaded, it's placed on the inverted microscope, a manual cell counter has been placed next to the microscope, and now we are focusing on the hemocytometer, being sure that we can clearly see the hemocytometer grid lines and the cells. Everything is in sharp focus, okay? Everything should be well defined. Then we're going to start to look at the cells and begin to count. And again, when we see live cells, we're going to push the red key. And when we see dead cells, we're going to push the white key. And those numbers will appear in the windows here. And if this unit is working correctly, the total number of cells will appear in the window over here. And once again, be sure that the counter is zeroed by turning these knobs. Be sure the counter is zeroed before you begin your cell counts. Okay, so again, this is what both chambers would look like in the hemocytometer, one on each side, okay? But we're going to focus on one of the two chambers. And then once we've counted in that chamber, we will proceed to count the opposite chamber. So we're going to focus on one chamber. We're going to, again, make sure that the grid lines and the cells are in sharp focus, that they're easily seen. Okay. Once again, then, let's remember what a square is. So this is a large square, one large square, and each large square is subdivided into 16 smaller squares. And this is where the cells will be that you are going to count, right? Inside of this large square. Cells out here will not be counted. Okay, and again, to be the most accurate with our cell counts and to get the best statistics, we're going to count a total of 5 squares per chamber, right? And with a little practice, you can get really good at this, and you can count 10 squares in 5 minutes or less. Okay, so there's our first, our second, our third, our fourth, and our fifth large square. And we're going to count the cells that reside within those squares. Nothing on the outside. Any cells out here, they are not counted. Okay? Okay. So, here's our square to count. We're going to look at the cells in here, right? So you're going to have to train your eyes to be able to count the cells in here, and hopefully these individual grids within the large square make it a little bit easier. But I recommend that you develop a consistent pattern and follow that pattern for every square that you count. So this is the pattern that I use, right? Left to right, then right to left, then left to right, then right to left. And so my eyes are constantly moving, and I'm making sure that I'm not counting the same cell twice or forgetting to count a cell. Some people go up and down, and that's okay. But again, the key is to make sure whichever pattern that you use, you do that same pattern in every single large square that you're going to count. Okay. In the previous slide, we talked about what's outside the large square and what's inside. So here you can clearly see, I hope, the cells that would be inside the large square. That's where we do the count. But we have the outside of the square, right? So that's the portion up here. Oh, there it is. So that's the portion up here. When you load the hemocytometer, there are going to be cells floating around everywhere, all right? So we need to be sure that we're only counting in here. So you kind of mentally block off those outside squares. Even though they're grid lines there, they're not within the region that we want to be counting, all right? And so that's going to be true for every one of these large squares that have an outside edge to them. So we do not count the area in red. We only count inside here. Okay, the other issue is that we can have cells that are on the line of a particular large square. They're right on the line. So if you look, they're right on this line, they're right on this line, they're right on this line, they're right on this line. Okay, so the question is, when do we count cells as being in the square that we wish that we are currently counting, and when do we count cells as being outside? And so the current convention is that cells on the left and cells on the top, if they are on the line, directly on the line, then they are counted as being in this square, right? And so the right here, the right for this square is the left for the next square. So any cells that are on this line will be counted as being in this square, and any cells that are on the top here will be counted as being in this square. And then similarly here, right, in the square below, if we were going to count the square below, the cells on this line, this would be the top line for this square right here. So any cells that are laying on this line will be counted as being in. Okay, so hopefully that makes sense to you guys. Let's see if we can illustrate this. Okay, so there's going to be my little hand-drawn cell, you know, a little oval shape with a darker membrane and a light gray interior. All right, so here we have some cells. Okay, these cells are outside of our counting square, and these cells are right on the line. So we are going to count these cells that are right on the line, right? And so I'm going to put a little green circle on them. The rest of these cells, they're fine, they're alive, they're happy, whatever, but they are outside the volume of the hemocytometer that we are counting. So we cannot count these cells. We just ignore them. We pretend they are not there. So we do not count those cells, okay? And then if we go to the left side, again, we have some cells that are directly on the line. So those cells we will count as being in this chamber. They're on the left line, so they're going to be counted as being in the chamber. So again, I'll put a little green circle in there. And the rest of these cells are clearly, none of them are touching the line. They're all on the outside, so they're not in the volume of the hemocytometer that we use for our calculations. So we will not count those cells. And again, that just illustrates our top and left lines. So hopefully that's clear to you guys. This takes a little bit of practice, and it also takes some concentration to make sure that you abide by that convention. Okay. Okay, so here is a picture of a cellometer with real cells in it. Right? I had trouble moving it into the field of view. This was the best image I could get. So this is not a complete large square. You see there are only 8 of the 16 squares here. But this was the best I could do. Sorry guys, but at least it allows you to see real cells. These are SF9 insect cells in a hemocytometer. So the questions that we want to answer here are which cells are inside and which cells are outside the counting squares. So this is the counting square, and here are the lines. So anybody outside the line, these cells are excluded. We do not count the cells that are outside the line, right? We do not count cells that are on the left line for this chamber, but the right line for this chamber. So what cells will we count? Okay. These are the cells that we won't count because they're outside the chamber. The cells that are on the line that we would count would be these cells. And maybe this cell, right? And so I'm going to go back and remove my little circle and question mark. This cell, you could count it in this chamber, or it's not all the way over on this line, so maybe you count it in this chamber. The key here is be consistent with your decision to count. So if you count a cell that's on this kind of line in the chamber here, you always do that. If you decide that you're going to count it in this chamber, you'll always do that. But be consistent with your counts, okay? And then there's lots of cells in here, so I don't want to draw circles around every live and dead cell, but the idea here is the cells where you see the outline of a membrane, but they are transparent, so to speak, at least in this picture. Those cells are live. Okay, lots of live cells. This is also a live cell. There's lots of other live cells, but I'm just sort of highlighting a few so you can see what they look like. And then the cells that are completely dark, right? Those are cells that are dead, so their membranes are compromised and they have taken up trypan blue. So those are our dead cells. And so this is where you will click on the white key, and with the live cells you would click on the red key. Okay? All right. Now, I cannot emphasize enough how important it is to get a good, sharp focus on both the hemocytometer grids and the cells that you're looking at. If you were trying to count from this image, your cells, your counts would not be accurate. Your counts would be incorrect. So make sure that you focus clearly on the hemocytometer grids and on the cells. Okay, now, again, this is a cellometer. We're looking at this hemocytometer. Actually, this is a real hemocytometer. We're looking at this hemocytometer under low power. We'd like to get it at a slightly higher power, but it illustrates several things here. So first of all, this is our counting square, right? So in real life, with not so good illumination, this isn't a very sharp picture. You can see it's harder to see the lines clearly, and the magnification is too low. We would want to go up to the next level of magnification. But that's our counting grid. So all of these cells outside the counting grid, we don't count these. These are completely excluded. Okay? Okay, the second thing here is that if you recall, we said the optimal number of cells to have within this grid to count is between 20 and 50 cells. There are too many cells here to accurately count. We need to dilute the sample. And again, when you regularly work with a specific cell line, CHO cells, 3T3s, SF9s, MDCKs, whatever, you should have an idea after several passages about what the dilution is that you need to make in order to achieve that 20 to 50 cells within a single square in order to get accurate counts. And the last reason that I didn't go to a higher power to focus was to look at this little piece of debris floating around in here. So this is either a piece of plastic that came from the manufacturing process of the slide, or this could just be a piece that came out of the Trypan Blue. But whatever it is irrelevant to the count. It shouldn't be there, but occasionally you see things like this, and it is irrelevant. We're not going to count this. This is clearly not any cells. And I guess maybe the other thing that we notice is there are some clumps of cells here, and we'll talk about that on the next slide. So here I've just sort of made a cartoonish picture of one side of the hemocytometer, one counting chamber, and what I'm going to ask you to do is look at this, and for just square number 1 and square number 2, determine the number of live cells, the number of dead cells, the total number of cells. I want you to really just sit here and look at this and see if you can count the number of live cells and dead cells, pretending this is under a microscope. I recommend that you do this at least 2 to 3 times, right, and see if you come up with the same number each time. And if somebody is viewing this with you who's in a cell culture class or who is a colleague at a company, have them do the count too, but don't share your counts with each other till you're done. So I'm going to count to 3 and pause this, and then count to 3 again and turn it back on, and let's see what we get. Okay? Okay, I'm back. So hopefully you've paused this. I'm back. We're going to count square number 1, right? And we're going to count square number 2. Okay. So if we look at square number 1, first let's look at this. We have a cell clump, the dreaded cell clumps, okay? These sometimes are unavoidable. They just happen, okay? So when you have clumps of cells and you're doing counting, there should be a statement in the SOP, in your cell counting procedure, about how to deal with clumps, right? And generally, there are 3 ways that labs or company labs deal with cell clumps. One is to count it as a single cell. So you look at this clumps of cells, you know there's lots of cells in there, but you just count that clump as if it were one cell. Okay, and again, the idea here is consistency. The second way that people sometimes deal with cell clumps is, especially if you can distinguish individual cells, if the resolution of your microscope is good enough that you can see at least some individual cells, then you count as many individual cells as you can and include that as part of your cell count. All right. Neither one of those ways is very accurate or very reproducible, but if that's what the SOP says, then that's how you handle a cell clump. The third way is to get a new sample of cells and try to disperse any clumps that are in that sample. So generally you would do that by triturating, by drawing the cells up and down gently in your micropipette, and as you squeeze them through the tip, hopefully the clumps break up. Sometimes that doesn't work, sometimes it does. But you have to have a method to deal with cell clumps. Okay, so let's go back now and do our counts, right? And again, we're going to have a pattern in which we count the cells, okay? And here what I got were 18 live cells, 2 dead cells, right? The 2 blue cells. This blue here is just an artifact. So in the real world, this might be a crystal of trypan blue that wasn't dissolved, or this might be a disintegrated cell, but it's not clear enough that we can say that this is an actual dead cell. Okay, so square number 1, 18 live, 2 dead, 20 total. Square number 2, I ended up with, again, 18 live, 3 dead, for a total of 21 cells. And if you're doing things right, the counts between the squares should be pretty consistent. They may be off by, as many as 2 to 4, maybe even 5 cells in total numbers, but they should be pretty close to each other. And then when you count the other chamber, when you reverse this and count the other chamber, again, the number should be pretty close to each other. Okay, so from here, we've learned how to count the cells, we've learned how to get the numbers. The next segments will deal with calculating the cell density, the cell viability, and seeding, how to do seedings. So that's the end of segment 3.